get on to some new stuff. Before we do that, I mean, I'm a big fan of this diagram. If I were you and you're, obviously we'll say this won't come up on your year 12 exam, which I'm predicting will be done end of July or 1st of September. But when you're there preparing for year 13, I would be drawing this out as some kind of huge poster, putting it on the ceiling above your bed or something. Read it last thing at night. It's got everything this. So let's see where we're up to. The only thing I would change, they just put H here. I'd put NADH there, to be honest with you. So far in this process, the glucose has come through the cell membrane into the cytoplasm. We've used up an ATP to make glucose phosphate. And then we've made 2 triose phosphate. We used another ATP. And then we've made pyruvate and we've yielded ATP. And to be honest, that looks like we've yielded the same that we've used, but that is not the case. And we've yielded some hydrogen. Now, I don't want to spend too much time talking about anaerobic, but it's worth pointing out that in anaerobic respiration, we form lactate in animals and bacteria, and ethanol if we're plants or fungi based. And that's going to occur in the cytoplasm. All of this next bit here that we're going to look at is dependent on there being oxygen here at the electron transport chain. If there's no oxygen here at the electron transport chain, it all backs up and the whole thing, this is inside the matrix, remember, this thing is all going to close down in the lack of oxygen. Quite a dangerous situation. And so we'll go this way in anaerobic respiration. It'll occur in the cytoplasm, and it will yield 2 extra ATP. So doing anaerobic respiration will yield around 4 ATP. How many do you think? Well, I had a guess. I don't like to guess. None of you know unless you've done extra research. How much do you think how many ATPs from one glucose molecule do you think all this palaver is going to net? It's going to be more than 2 that we've got here. Let's have a guess. Someone have a guess. Oscar, how many do you think?
Let's go for 6.
Yeah, you say aerobic respiration is better than anaerobic. Aerobic's producing 2. This will probably be 3 times better. Probably yields 6. It's the wrong answer, folks. It is 32. Okay, so all of this is designed from one glucose molecule to make 32. The second stage, so let's just look on the diagram what we're doing. In outline, we're going to get pyruvate, it's going to give off CO2, it's going to give H in the form of NADH, and we're going to end up with acetyl coenzyme A. Okay, and this is the link reaction. Let's get straight to it. So the pyruvate, let's remember where it's located. That was made in the cell cytoplasm. It's going to cross the first membrane of the mitochondria. It's going to cross the second membrane of the mitochondria, and it's going to go into the matrix. So we are now in the matrix. It's great, isn't it? We are in the matrix. And so there's a couple of words you need to know, like decarboxylated is going to happen. And all that means is that CO2 is removed from the pyruvate. And there's a pattern here that every time you see CO2 removing, you'll see NAD become reduced NAD or NADH. Okay? And then we get something unfortunately named acetate. I'll say, well, that's unfortunate in a minute, but the point you need to remember, it's got 2 carbons. And then that acetate is going to bind with the coenzyme A. So remember there were enzymes and coenzymes are taking the product away from the enzyme. okay, transporting it away from the enzyme. So all of these categories, as I said, require enzymes and quite complicated enzymes. So pyruvate is oxidized, NAD is reduced, just as triose phosphate. Now, so what I'm going to show you is for the chemists, really. We've got acetate here. Now, you know, years ago, ethene used to be called ethylene. Ethanoic acid used to be called acetic acid. All right. And then they had a big standardization of naming, nomenclature, to standardize it worldwide. And the memo never reached the biologists, and they never bothered changing to the new. And there's still some older chemists who will refer to, or better not ask what Dr. Ray refers to, acetylene instead of ethene. But everybody was supposed to switch, and what that means is that we should not be calling this acetate. That should be called ethanoate. Okay, and so those of you that know anything about chemistry should know what ethanoate ion looks like. Okay, does that sound familiar, chemists? Ethanoate ion.
In the biology exam, should we call it acetate or ethanoate?
Yes, you've got to call it acetate. Nobody calls it ethanoate. It's got to call it acetate. And the reason is because then because there's all kinds of related chemicals. Like if you start calling that ethanoate, you're going to have to call that ethanyl coenzyme, and just nobody calls that ethanol. So, but for those of you interested, that's actually, again, the pyruvate. It says that's pyruvic acid there. Actually, to be clear, you're just going to call it pyruvate. We're going to get dehydrogenation and decarboxylation. So CO2 is removed from there, and that's why it's going from 3 carbons to 2, obviously, and an H is going. And so we're left with the ethanoate ion, which you must call acetate. And if you want to know what acetyl coenzyme is, the coenzyme there has replaced the O minus, okay, and is going to transport this vinegar, this acetyl to the Krebbs cycle. Okay. So yeah.
Why does the hydrogen in the pyruvate have a plus on it?
That's because the formula for pyruvate, it doesn't have a hydrogen there. Okay, but if I showed you that, you're going to say, well, where's the reduction from NAD gone? And so what it means, it must actually be pyruvic acid at this point. And so that is something. So without the H plus, it's pyruvate. With the H plus on it, it becomes pyruvic acid. Okay, but unfortunately, it seems to be people don't really distinct. You just call it pyruvate all the time. Okay, but I cannot see how that can reduce NAD if it didn't have a hydrogen on it, but we'll see. There's perhaps a reason for that. But the point is, you don't need to know all these structures. I'm putting these structures because I actually find it easier to visualize these structures alongside the words. I can see 3 going to 2 plus an enzyme. I can visualize that better. For God's sake, don't try and. We've talked about the enzymes being complex. The reaction mechanisms in here is third year degree level. So we're not going to look. Please don't try and work out. Oh, I wonder how we lost CO2 here. What attacked what? You literally just need to know the words pyruvate, acetate, acetyl coenzyme. But in case you're interested, that's ethanoic acid or ethanoate ion. Okay, you could quite happily study all this and never realize that that is that, because the biology books won't print it. So that is the link reaction. That's the main. Sorry, I should have just said a couple of things. Yeah, a couple of things I need to say about that before we go on. So obviously, this pyruvate dehydrogenase complex is the enzyme. Okay, that catalyzes that. It's quite a monster enzyme. You don't need to know anything. Whereas NAD is the coenzyme. Okay, so that means that the enzyme is catalyzing the reaction. The coenzyme is taking away the product from the enzyme. So the product doesn't stay in the region of the enzyme. And then we have this second coenzyme A this time. This is to transfer, to bind with the acetate, and it's going to transfer it to the Krebs cycle. There's no ATP here, okay, and there's no NADH here. Alrighty then. Shall I move it on? I think the next slide is very similar. Woo. No. So the next slide is very similar. It's just pointing out for every 1 glucose, 2 pyruvate molecules will be made, okay. And so 2 acetate molecules will be made, and 2 acetyl coenzyme A molecules will be made. So this reaction is happening twice per 1 molecule of glucose, but obviously because the glucose molecule is split into 2. Okay, so the reason you'd want to know that information is if you want to say, well, how many ATPs were made for 1 glucose atom, or there's 2 ways of looking at this. How many ATPs are made from 1 glucose, and the other way of looking at it is how many ATPs were made for 1 cycle. Okay, there's 2 different questions because you'll get 2 cycles for each 1 glucose molecule. So this is still a recap. So the link reaction makes 2 acetyl coenzyme A molecules. So that was the ethanoate ion combined to the coenzyme, and that's going to be transported to the Krebs cycle. Now, this is assuming from 1 glucose, okay? 2 carbon dioxide is given off as a waste product, 2 reduced NAD, and they are going to go to oxidative phosphorylation, okay, which was that final step occurring in the membranes where you're going to get your 32 ATPs made. All right. So every NADH or reduced NAD that you make is going to end up useful for making ATP eventually. Okay, so every time we make a NADH, we're very happy. Okay. So here's a load of reactions we need to look at, and luckily for you, the A-level syllabus simplifies this process, as it'll become apparent. So the acetyl coenzyme A there is going to form citrate, which is a 6-carbon molecule. So how can we form a 6-carbon molecule? Well, we're going to use this chemical, you need to remember the name, oxaloacetate, which is a 4-carbon. So this is obvious. You add a 4-carbon molecule to a 2-carbon molecule, and you're going to get a 6-carbon molecule called citrate acid. So this is sometimes called the citric acid cycle. The coenzyme has done its job. It's brought the acetyl to the Krebs cycle. It's going to go back now to the link reaction. Okay, so it's reusable. We have formed citrate. Okay, now don't get too hung up on this, but obviously 4 plus 2 is 1, 2, 3, 4, 5, 6. Okay, so oxaloacetate plus acetyl coenzyme ends up as citrate, and this coenzyme A is going to detach and go off. So you just need to remember the word equation and the number of carbons. That's it. Okay. This next stage now, citrate, 6 carbons, is going to go to a 5-carbon compound. We don't even have to remember the name. I think it might be malate or something like that. We don't need to remember it. And so obviously for it to lose 1 carbon, how's it going to do that? It's going to produce waste CO2 here. At the same time, NAD is going to be reduced to NADH, okay? And so the equation we're trying to remember here is, it's worth mentioning again that whenever CO2 is given off, this is called decarboxylation, okay? Carbon dioxide is removed. It's also worth pointing out every time hydrogen is removed from a molecule, this is called dehydrogenation, okay, it's a technical term. And each time we do dehydrogenation, we're going to produce NADH, because NADH will yield us ATP later on. And so that's the equation you need to remember there. Citrate, C6C, goes to a 5-carbon compound. So that's much simplified. Probably don't get bogged down with mechanisms, but you'll see something's going on around that carbon to reduce it down. Okay, now I said it's a simplification. We're now going to go from this 5-carbon, this nameless 5-carbon compound, to oxaloacetate, which is a 4-carbon compound, and there's quite a lot going on here. And what that tells you is there's loads of other chemical reactions going on here that you would need to memorize if you went to medical school or did biochemistry. You don't actually remember all of these. I don't know why, because you can look them up, but you'd have to memorize them. So there's a lot more reactions going on here. So what do we need to know? Another CO2 is coming off, and every time CO2 gets given off, we always see a NAD going to NADH. We're going to have an ATP made during this sequence of reactions from ADP, so that's all good. And then you're going to get a different coenzyme called FAD appear, and that's going to be form reduced FAD. And then we'll have a second NAD forming reduced NAD. Okay. And so this is called the regeneration of oxaloacetate, because we've got that 4 carbon again, ready to react with 2 and go back into the cycle. Okay. So we're basically feeding new 2 carbon blocks all the time, and then we'll be breaking it down to 4 carbon blocks. And it's feeding with another 2 carbon blocks, etc. So it's going around in a circle. In terms of the numbers, keep an eye on them. 1 reduced NAD there, 2 there, 3 there. 3 NAD H. Bless you. 3 NADH produced. 1 FADH produced, 1 ATP produced. CO2, 1, 2 CO2s produced. Okay, so, and that's it for CO2. If you think about it, we've got 1 produced there, 1 produced there, 1 in the link. So, is this difficult? No. Is it a lot to remember? Yes. I mean, it's literally, as I say, if we're in lessons, I'll be making a poster of this or writing it out neat, and then try and write it out. You'd try and write it out neat, and then you'd close your textbook and you'd try and write it out. You'd have to write this out from memory would be the only way you'd test yourself. Can I write this out? Can I write this cycle out from memory? Without looking at the book. and including key factors like the number of carbons, 654, the actual names, citrate, don't need to know malate, oxaloacetate, where the CO2 is given off, you have to get that right, where the NADs are given off, you have to get that right, and then the FAD. I say this is greatly simplified, so that's a help. Okay. 5 carbons there going to 4 carbons there. It's a hell of a lot more complex. You know, you can see every one of these is a chemical reaction gone on. So we're just going to summarize that. Okay. So for one glucose molecule, we'll have 2 turns of the wheel or the cycle. So we'll get 2 coenzymes A, 2 oxaloacetate, 4 carbon dioxide. Remember I talked you through one cycle, had 2 carbon dioxide, 2 ATP, 6 reduced NAD, and 2 reduced FAD. Okay, that's per one glucose molecule per 2 turns of the cycle.